Hey, good morning, everyone. So today we are reviewing for our second exam, which will start on Wednesday. So remember, just like the first exam, this exam is broken into two parts over 2 days. So this Wednesday, October 20th at 10:30 a.m., so our normal class time, all you have to do is log on to Laulima. You do not use Zoom at all. Just get onto Laulima, and at 10:30, the multiple choice part of the exam will become visible. I think up until that point, it won't show up. So at 10:30, if you don't see it, just hit refresh in your Laulima, and you should see it. Just like last time, the exam is open for an entire hour until 11:30. So it's up to you when you actually click begin to start the multiple choice part. Once you click begin, you'll have 50 minutes to complete it. So again, that means that just as long as you click begin and start by 10:40, you'll get your full 50 minutes before the exam shuts down at 11:30. All right. And then later in the week, either Thursday or Friday during your normal recitation time, go and attend recitation in person, and you will take the free response written part of the exam. Okay, so it's the exact same format as last time. All right, let's go ahead and take a look at the topics that you're responsible for. So you are responsible for lectures 12 through 20. And over the course of these lectures, there were two main topics that we were discussing. One is gene regulation, in other words, how living cells decide what sets of genes to actually go ahead and express. And remember, that has a lot to do with, many, if not most genes in the genome are genes that need to be expressed because they represent the creation of proteins that are needed for everyday sort of generic living functions inside of cells. But then there are also subsets of genes that maybe code for proteins that are more specific for certain functions that some cells might need, whereas other cells, it might be completely irrelevant to have that protein made, right? And so we took quite a bit of time understanding the different levels at which a living cell can have control over what subset of genes gets expressed. The second main topic in this group of lectures then is understanding the function and the structure of cell membranes. And so that's what we wrapped up talking about last week. All right, so here we are in week 9. Today's our review session. And again, Wednesday is the multiple choice part that you just take remotely from wherever. Just get on Laulima and make sure to remember to take it. And then later in the week, go to recitation and take the written portion of the exam. Okay, so let's go ahead and review. So let's sort of quickly go through examples of the different levels or layers of a cell controlling things like gene expression. So we started off discussing things like bacterial or prokaryotic operons as an example. Now, I know when learning about these operons, it can be kind of confusing. And you might say, well, I don't understand why we learn about how bacteria regulate these genes because it seems like such a specialized, example. The reason we do it is because in many ways, these bacterial operons are sort of like simpler versions of things that happen in our eukaryotic cells. Right? And so if we use these operons as examples, we can understand certain concepts, right? Important concepts that still apply to what goes on in, for example, a human cell. Right? And those concepts have to do with the fact that you're talking about genes being sequences along a strand of DNA, right? And whether the cell, whether it's a bacteria, for example, how does it decide or have control over whether those genes are expressed? Right? Part of that is understanding the role of different proteins that are involved in making these kinds of decisions. and importantly, sort of conceptually understanding that gene expression oftentimes has a lot to do with whether that particular gene's product is actually needed at a given moment by a cell, right? So that's why these operons are good examples of that. For example, we started off looking at the Lac Operon pictured here, right? So this is the set of genes, set of 3 main genes in something like E. coli, for example, E. coli bacteria, that code for proteins that are involved in the breakdown or catabolism of lactose sugar. Right? So we said normally, if you have a bacteria that's maybe sitting in a puddle of water by the side of the road, there might be very little lactose sugar in that environment, right? So why would the bacteria want to bother using valuable resources like amino acids and things like that to make proteins that are involved in lactose catabolism if there's no lactose around, right? It wouldn't. So most of the time, we said that this Lac Operon would be constitutively repressed. Remember, constitutively means sort of like the default state, right? So here, that involves a protein, this red repressor protein that's coded for by some other gene upstream, like this LAC-I gene, right, that ends up transcribing, or when it's transcribed, it makes this LAC-I mRNA that gets translated into this repressor protein, and this repressor protein is by default the right shape to bind to the promoter region at this operator sequence, thereby blocking RNA polymerase from coming in and transcribing these 3 lactose pathway genes, right? Again, because if there's no lactose around, the cell doesn't need to worry about making these products. However, in the presence of lactose, so suddenly this E. coli is not in a puddle by the side of the road, it's actually in a carton of milk, for example. So obviously, right, this living cell needs to be able to adapt and take advantage of different types of nutrients that are suddenly in its environment. So lactose, the green L here, right, it will bind to that repressor protein and prevent the repressor protein from blocking the promoter region of the Lac Operon. And as a result, right, RNA polymerase, the big purple thing, can come in now and start transcribing. And so that RNA polymerase ends up making this RNA, this red thing, right? Because here's the RNA polymerase kind of heading down the set of genes here. And in the case of this Lac Operon, different parts of this mRNA get translated separately to make the 3 protein products, right? That can now go to work and start breaking down the lactose to get the glucose out of it, which is what the E. coli or the bacteria wants to use, right, for energy. So we looked at this as an example of a constitutively repressed operon, but we also have examples like the tryptophan operon, right, where the product of the operon, in this case, the amino acid tryptophan, is always needed in the cell. So therefore, the trip operon would be the opposite. That would be constitutively active, right? Until, for example, maybe you have too much tryptophan compared to the other amino acids. So the cell says, I don't want to waste resources making more tryptophan. I could use those resources to work on the other amino acids, right? In that case, then the trip operon is shut down, right? So you have things like a feedback loop here with the Lac Operon, right? That's an activating loop by the lactose that activates transcription, whereas in the tryptophan operon, it would be the opposite. It would be an inhibitory or repressive loop, right? If you have too much tryptophan, it ends up inhibiting the production of more. All right, so go back, take some time and understand those examples and realize, right, what I hope you guys realize is it's not that I think these bacterial operons are like the most amazing thing ever or something like that. No, I've been trying to use these to teach you guys some simple concepts that still apply to more complicated organisms like us and how our cells regulate our genes. All right, so we said that there are 5 levels of gene expression, each one representing a different opportunity for a living cell to control whether you actually end up getting a finished functional protein product from a gene. Right? So we looked at, for example, genomic control, for example, at the fundamental level of the DNA and the chromatin itself. Right? By adjusting how chromatin is structured, you physically are controlling whether certain stretches of DNA are exposed or available, right, to be transcribed. Right? Then transcription, we looked at different examples of how transcription factor proteins that actually bind to the available or accessible DNA need to assemble in order for them to have a cooperative effect of doing things like eventually recruiting RNA Pol2 to the promoter of one of our genes so that transcription can happen. Then we learned how when the RNA is coming out of the RNA Pol2, it undergoes editing. Right? So the primary RNA transcript that has basically a perfect copy of the gene that was being transcribed, but now in RNA form, right? How it gets processed so that you can have then a finished mRNA or messenger RNA that represents the particular version of this gene, this gene's instructions that the cell now wants to convert or translate into the actual protein product, right? And so translation then, step 4, is taking place with ribosomes that are translating this pink messenger RNA and making this squiggly purple line, which is a chain of amino acids that together, finally, when it falls off, can then fold up into the functional protein. And then step 5 we looked at was even once you have the finished functional protein, right? Post-translationally, meaning translation has already taken place and you have this polypeptide sequence, it can still be modified, right? In ways like adding little chemical groups or whatever to change its shape, right? In order to, for example, switch it on, switch it off through alterations in its tertiary structure, right? So I recommend going back and using this slide as a good way to review. What I recommend you do is look at the examples that are listed in the flowchart on the left, right? And actually make yourself look over at the illustration on the right and try to kind of match up where these different things are happening, right? And use that as a guide, because then you would then expand upon understanding the details by looking at our lecture slides and study guides for that particular set of details at that level. Okay. All right. So let's go through some of these. We said at the fundamental level of regulating chromatin structure, right, making the chromatin more compact or heterochromatin-like, right, inhibits gene expression because you are quite literally hiding or concealing the DNA sequence, right, from the proteins, from the transcription factors that are need to bind there in order to express it. We said that one way that the DNA itself can be modified, right, is through the addition of methyl groups on cytosines that are part of CpG dinucleotides. Remember, CpG dinucleotide means you're referring to a C next to a G, right, on the same strand of DNA, right? Not C's and G's across the middle of the DNA double helix across 2 strands, right? You're just talking about the C's next to the G's on 1 strand. We said that promoters are often enriched with these CpG sites. And in fact, many CpGs lined up together can form almost like a CpG island, right? And this is where enzymes like DNA methyltransferases get recruited, and they put methyl groups on the cytosine, making them 5-methyl cytosines. So in this figure on the left here, we see that the thick black line is the DNA wrapped around nucleosomes, and the red dot here, right? You can see the red dot is actually on the black lines themselves because those are actual CpG sites in the DNA that have gotten methylated, right? And as a result, that methylated DNA ends up being compacted because both the nucleosomes can hang on to the methylated DNA tighter, and you have the recruitment of different proteins that do things like methylate the tails of histones, right? So the red stars are histone methylations. So keep in mind, it's two different things, right? Methyl groups are methyl groups, right? They're just CH3s, but you can add them either directly to DNA, thereby chemically modifying the cytosine in DNA, or you can add methyl groups to amino acid residues like lysines, for example, that are part of the histone proteins that have their N-terminal tails sticking out, right? In either case, you're adding methyl groups, but it's important to realize you're either doing it to the DNA directly or to the histone proteins, right? And that results in the chromatin being very compacted, especially if you have these CpG islands in the promoter, right? And that would really discourage transcription factors from being able to recruit RNA polymerase to the promoter, right? On the other hand, unmethylated DNA, so instead of the red circles, they are empty white circles, right? Favors the DNA unraveling from the chromatin, right? And it also then results in the recruitment of histone acetyltransferase proteins that acetylate or acetylate the histone tails, opening up the chromatin, making it more transcriptionally available. Okay. So then we looked at how if you have a core promoter region over here, and let's say you have maybe this whole region close to the green region of the promoter contains other elements that help with the transcription factors piling up, the general basal transcription factors piling up, recruiting RNA Pol 2 here, right, to the core promoter. This darker blue region would be the actual coding sequence of the gene heading off to the right here. Right? We said that many, if not most, genes also have other regulatory sites that don't necessarily have to be close by. They can be surprisingly far away on a strand of DNA, tens of thousands of base pairs, either upstream or downstream, right? We said that those sites, for example, enhancer sites, are places where you'd find DNA binding motifs in the DNA, short sequences that particular proteins, like these green activator ones, recognize by their shape, right? And bind there and start building up other transcription factors like histone acetyltransferases, for example, and they start piling up at these kind of staging areas that are kind of remote from where the actual gene is, right? So that's why we call them distal or far away. Eventually, right, the piling up of transcription factors facilitates the interaction, the physical interaction of this distal regulatory site, like this enhancer, right, and actually physically brings it to close proximity with the core promoter so that all of the transcription factors that were piling up on the enhancer, their activities, right, can be directed now to the promoter. Right? And that's why the reality is it doesn't matter that this enhancer is upstream of the gene or downstream of the gene, because in 3 dimensions, right, something being upstream or downstream on a strand of DNA is meaningless because the DNA is just all wrapped around. Okay, so when we discuss transcription factors, we said it's very important to understand, right, that the specific subset of transcription factors that might be more specially expressed in different sort of levels, low versus high, in particular cells, plays a very big role in determining how different that particular cell type is, right, compared to another cell type that might have different expression levels of those transcription factors, right? Or maybe one cell type expresses that protein from the gene, right? And some other cell type simply doesn't express any protein from the same gene. So go back to this example here, where we discussed that if you thought about 3 different, very different cells in your body right now, right? Skin cells, nerve cells, and maybe red blood cells that are maturing in your bone marrow, getting ready to go to work in your blood. We said that many transcription factors would be common among these cells because, after all, many genes, if not most genes in your genome, right, are there because they make some critical component that all living cells need, right? So that would be like this, I don't know, this transcription factor here, the one that's kind of a pinkish, looks kind of like a little bear claw or something over here, right? But we said that other transcription factor proteins, right, that are coming from genes, or I should just say any proteins coming from another subset of genes, might be quite different, right? So we said that the nerve cell might express this yellow tooth-looking one. The developing red cell might express this red diamond one, right? And as a result, if you take the same gene in the genome of these three cells, because remember, genomic equivalence means all the cells in your body have the same human genome in them, your genome that you got from mom and dad, your genome that mom and dad gave you, right, the moment of conception, when two copies of the human genome came together, one from dad, one from mom, and every single cell in your body now, trillions upon trillions of them, 20-something years later, right, have that genome in it. So this gene that we're looking at in the cartoon, right, all of your cells have, but maybe this protein that's encoded for by this gene, right, is a bit more specialized. The function of that protein. Maybe some cells need it, some cells don't. So we said in the skin cell, right, at the core promoter of this gene, yeah, you would have general transcription factors, again, examples of proteins that all cells make from the genes that make these transcription factors, because these are all just critical, normal, generic transcription factors needed to turn on any other gene. Right? So they would be like these purple spheres. And of course, RNA polymerase is another example. All of the cells in your body have to make RNA polymerase proteins, right? And all the different subunits that make up the RNA polymerase. So those would come together on the promoters of this particular gene here. Maybe in the skin cells, right? They make a moderate level of the resulting protein that this gene codes for. Maybe in the neurons, right? Maybe neurons need to have a lot of this particular protein. So because the neuron had this yellow tooth transcription factor in it, it binds to the DNA binding motif that's at this gene, right, in neuron cells, and helps really, really kick up the expression levels of the protein there. Whereas we said maybe in a developing red blood cell, maybe red blood cells, they don't care about this protein. They don't need it at all, right? And so the red blood cell might have this red diamond transcription factor that binds to the red DNA binding motif. Right? And as a result, in the developing red blood cells, maybe it represses the expression of this protein. Right? Keep in mind that this gene is identical in all three cells. They all have where the yellow tooth-shaped transcription factor would bind. They all have where the red diamond-shaped transcription factor would bind, right? The difference is some cells have those transcription factors in them, some cells do not. Right? And I asked you to consider that as a result of this protein product being made in very different levels in different cells, if this blue pentagon itself is a transcription factor that will bind to the DNA binding motifs that it recognizes surrounding different genes in the genome, right? Then you'd have the next wave of protein expression, right, as a result of the activity of this blue pentagon protein, right, pushing the difference between these cell types, skin, neuron, and red blood cells, further and further apart. Right? And think about going backwards in time, all the way back up, right? Because after all, the red diamond and the yellow tooth-shaped protein themselves came from genes, right? And in red blood cells, those genes, right, favored production, or those genes were activated to make the red diamond, right? And in the neuron, the genes that made the yellow tooth were the ones that were preferentially expressed. And if you go backwards in time like that, you go all the way back to the original pluripotent stem cell. Right? And so that was the example of the origami that I used. Remember, at the moment of conception, the pluripotent or totipotent stem cell, right, is like this blank square of origami paper. right? No decisions have been made yet. This piece of paper can literally become any origami animal, right? Just like that fertilized human egg, right, is going to become any type of human cell that makes up the body. But once you start making decisions, and those decisions, are not physical folds of the cell, of course, but each fold of the origami paper represents a wave of genes that get turned on, right? And then the products of those genes that got turned on themselves will differentially control what the next wave of genes are that get turned on, right? Based upon where around genes these different transcription factors bind. Right? And with each fold, you get further and further down the path of committing to a particular origami animal or committing to a fully differentiated cell type among the 220 different cell types you have in your body, right? Okay, so keep that in mind. Remember, in our adult cells, in our adult bodies, we no longer have pluripotent stem cells anymore, right? What we have in the different tissues of our body are these multipotent stem cells. In other words, stem cells that can still become one of several different types of tissue-specific cells that these multipotent stem cells are meant to constantly replenish, right? However, because these multipotent stem cells are already far enough down on this decision-making tree of gene expression, right? If this were, let's say, a hematopoietic stem cell in our bone marrow that can make one of several different types of blood cells, right? That's great, but that hematopoietic stem cell cannot make something like a nerve cell or a skin cell. Why? Because those decisions, right, were farther up in the decision-making process, and those cells had already branched off, before then, depending upon their decisions that led to other groups of cells that they were destined to become. So we examined how when RNA polymerase starts transcribing, right, this is another example of an opportunity for controlling expression. We said that the C-terminal domain, or CTD, of one of the protein subunits that makes up the big RNA polymerase holoenzyme, its amino acid sequence in this tail region is a set of repeating amino acids. Most importantly are the serines, because these serines can be post-translationally modified with phosphate groups. And depending upon changing patterns of which serines end up being phosphorylated, that will change the shape of this C-terminal domain of the RNA Pol II and attract different sets of proteins. For example, as the little red RNA first starts coming out, the phosphorylation pattern of the serines in the RNA Pol II CTD is recruiting things like capping enzymes, like this orange or this yellow thing here. And this capping enzyme then is brought close to where the new RNA is coming out, so it can go ahead and put the 5' cap. Along the course of the RNA going down the gene, as the RNA, resulting transcript is getting longer and longer coming out of it, right? The shape of the C-terminal tail changes by changing patterns in serine phosphorylation. And then you recruit things like the splicing machinery, for example, that then goes to work and starts editing the RNA. So the result of this, right, is you'll have a pre-RNA, you can call it a pre-mRNA or just a pre-RNA that has these different components in it, right? 5' cap, poly A tail, and introns and exons that can be edited. Right? So then even when you have the finished mRNA, there's still an opportunity for your cells to have some control as to whether or not that finished messenger RNA actually gets translated by ribosomes or not, right? So the example we looked at was how mRNAs of iron managing proteins might be utilized in response to things like the presence or absence of iron. We looked at two proteins. Ferritin is a protein inside of cells that helps to store, right, store extra iron. So in this case, normally this ferritin mRNA, right, would have a some kind of a large purple protein here, which is a iron response element binding protein. Normally, it would be docked on this particular sequence within the mRNA. Keep in mind, right, this sequence, this IRE, right, is just simply a sequence of RNA, right? That's the right shape, the right sequence for this purple IRE binding protein to bind to. So normally, if there's no iron, this IRE binding protein would be docked there, and it would get in the way of ribosomes translating this mRNA. Right? But once you have a lot of iron present, the iron binds to the IRE binding protein, changing its shape, and then the purple IRE binding protein falls off of the mRNA, as pictured here. And as a result, ribosomes can now go ahead and start working their way down the mRNA, making the ferritin protein. Right? So the ferritin protein is made, and then your cell uses that ferritin protein to store all this extra iron that's floating around. Right? On the other hand, getting iron into the cell in the first place is the role of the transferrin receptor. Right? So normally, if there's little iron in the cell, your cell wants to get more iron into it. Right? And so it would need to have these special proteins on the outside that receive transferrin molecules that are carrying the protein around in your blood, or are carrying the iron around in your blood, right? And encourage these transferrin molecules to come into the cell to bring the iron. Right? So normally, right, this mRNA of the transferrin receptor would be actively translated, right, in the absence of iron, so that you can make the transferrin receptor and get some iron into the cell. And the way that works is this mRNA also has an iron response element sequence in it. In this case, the particular IRB binding protein. Keep in mind, this example of an IRB binding protein would be a different protein than this one up here, right? They're just using these as sort of generic examples of iron response element binding proteins, right? The IRE sequences themselves in these two cases would be different, right? Because IRE doesn't mean it's a specific sequence, right? All it means is it's some kind of sequence that allows translation of this mRNA to be influenced by the presence or absence of iron, right? In other words, translation is responding to the presence or absence of iron based upon whatever these sequences are in the IRE region, right? So normally you would have this IRE binding protein bound to this IRE over here, right? And that would prevent these different degrading enzymes from cutting up this mRNA, right? So as a result, normally you'd have ribosomes coming in at the start codon, translating, and then stopping at the stop codon, and you'd be making the transferrin receptor protein. Right? But if you have now a ton of iron in the cell, right, the cell no longer wants to waste effort or resources making more of the transferrin receptor on the outside, right, because they just keep bringing more iron in. So the presence of a lot of iron ends up releasing this IRE binding protein from the IRE here, and now these enzymes can come in, these proteins, and they can cut up the mRNA, thereby stopping more translation from happening. Okay, so you can see, even though these sound very complicated, and this is what's happening inside of our cells, right? It's really still many of the same themes as the operons in bacteria. Right? So that's why it's important to talk your way through these and really try to understand what these examples are trying to tell you about how genes are often regulated at different levels by proteins, and often by the presence or absence of whatever it is that these proteins are meant to work on, right? Like in this case, iron. Okay, so then we shifted gears and started talking about cell membranes. So we said, importantly, that the cell membrane of our cell is a phospholipid bilayer, right, with all kinds of interesting proteins kind of anchored to it. Some of these proteins are glycoproteins, meaning they're combinations of protein and short oligosaccharides or chains of sugars. Right? Many of these proteins serve as, for example, channels, right, or transport proteins that are in charge of moving things back and forth between the two sides of the membrane. Remember, it's fluid, because the main thing that's in there that's keeping it fluid are the phospholipids being held together by water, right? And that's one thing that I never want you to lose track of, is how amazing that is, right? The fact that our cells are held together by the fact that they're in water, right? And if it wasn't for the water, our cells would fall apart. That means our tissues would fall apart. That means our organs would fall apart. That means we would fall apart, right? We are held together by hydrophilic and hydrophobic water interactions, which is amazing and kind of terrifying if you think about it at the same time. Okay, so the three main lipid classes that make up cell membranes. So review the structure of a phospholipid, right? Review that we have glycolipids, right? And that also involve fatty acid tails. They may or may not involve a glycerol group, right? But they certainly involve some sort of carbohydrate or series of carbohydrates, maybe a short chain of carbohydrates, like an oligosaccharide, right? And sterols. So sterols are another example of a fatty acid chain that has sort of been folded up as rings, and sterols, like cholesterol, for example, play a very important role in being embedded in the phospholipid bilayer, and they help maintain the fluid sort of integrity of the phospholipid bilayer, right, by keeping the phospholipids sort of properly spaced apart. And we learned that sterols, like cholesterol, are very important for the synthesis of different types of hormones, like the sex hormones, testosterone and estrogens. Okay, so phospholipids, make sure you go back and review the structure, right? Remember, some key things are that a phospholipid is made up of two fatty acid chains held together by a 3-carbon glycerol. Right? 2 of those carbons in the glycerol are hanging onto the fatty acid chain. The 3rd carbon is hanging onto a phosphate group with some kind of variable R group also linked to it. Right? And so remember that that gives these phospholipids an amphipathic property of having one side, the polar side, that plays very happily with water molecules, and the nonpolar hydrophobic tails that end up getting pushed by water molecules away, right? And so just by its very nature, if you have a whole bunch of phospholipids like this, right, their tails will all end up getting pushed together as the water on the outside continuously works to shove them around. We also examined how you have different types of proteins embedded in the membranes. And I think an important thing to remember is, proteins like, for example, this globular protein here, right, that might be soluble. In other words, normally it floats around in the water as a blob with all these water molecules kind of dancing around it on the outside, right? Those amino acid residues that point to the outside would be the ones that are hydrophilic. But you'd have certain amino acid residues that might be hydrophobic. For example, in this globular protein on the left here, maybe this side of the polypeptide chain, when it's folded up, actually has a lot of hydrophobic side chains sticking out of its amino acids. As a result, water molecules will just shove that part of the protein into the phospholipid bilayer. Right? Same thing with these membranes that are transmembrane proteins that go through the membrane. Right? Think about the nature of the amino acid sequence of the portion or the domain of the protein that is meant to be anchored in the middle of the membrane compared to the nature of the amino acids that you would expect to find right on either side sticking out into the water. And then this is just a reminder that membrane proteins have diverse functions. Right? As part of being glycosylated, they can change their shape a lot. Right? Or in some of them, like especially the multipass units that zigzag back and forth, they can form 3-dimensional channels. Okay? So then we started studying how things move across the membranes. So remember, simple diffusion are small molecules that can just make their way through a membrane going from high to low concentration gradient. The membrane slows them down compared to if there was no membrane there. Right? But these molecules can just move through, being pushed by the concentration gradient. Some molecules that are larger, right, require help getting across the membrane. So then you'd have this large green protein here that is facilitating the diffusion through. Again, right? It's not requiring any energy because this big red arrow pointing down is being driven by the concentration gradient of these. Let's say these are sugars like glucose, right? On one side being high, the other side being low. So the natural tendency will be for it to flow downwards in this illustration, right? And that's facilitated by the fact that you need to have this protein here because these sugar molecules cannot make their way through simple diffusion through the membrane. And we learned that lots of times your living cell needs to, on purpose, push things against the concentration gradient, right? And that requires energy. And so you'd need this green protein here, some sort of a pump, for example, right, that needs to change its shape against the force of the concentration gradient of these purple squares so that it can continue shoving these purple squares from a low concentration side to the high concentration side. And that requires energy, oftentimes in the form of a molecule of ATP, right? So by breaking the ATP and the high energy bonds in the ATP, this transporter protein, right, it changes its shape. And when it changes its shape, maybe it shoves another one or two of these purple squares through it. Okay, so examples of active transport, we looked at things like ion pumps, like the sodium-potassium antiporter, right? So in this case, we learned that cells can pump 3 molecules of sodium from inside the cell to the outside, right? And then as this protein changes its shape, right, it can then release the sodiums to the outside and also bind to potassiums on the outside. And then when the protein changes its shape back, it'll actually bring those 2 potassiums into the cell. And we learned that that's how our cells on purpose maintain this very strong disequilibrium of sodium and potassium inside the cell intracellularly versus outside. Okay. Remember that oftentimes the pumping of these ions to set up different gradients, like low potassium on the outside and high sodium on the inside, is done on purpose to set up gradients that can then be utilized to drive other transport. So go back to the lecture, look at the cartoon that shows like this antiporter, in sort of a circle, right? I think I don't know how to better describe it. Like the cartoon is a circle showing different steps of how this antiporter works, right? And talk your way through it. Why is this important, right? What is happening along the way? And importantly, look at how ATP is used. All right. So oftentimes our cells set up these intentional concentration disequilibriums, right? These gradients in order to tap into it and make use of it to drive other things. So a good example is proton gradients. So here we have on the left, this purple protein is a direct transporter or a direct ATP pump that pumps protons from inside of the cell where the concentration is very low out to the outside of the cell where the concentration is actually very high, right? So since you're going uphill in terms of going from low to high, this requires energy. And if this transporter protein does this directly by breaking ATP to change its shape to do that, that's an example of direct active transport. But the protein on the right, right, is also an active transporter, but it doesn't at all involve the breaking of ATP itself. Right? What it does is it takes advantage of the fact that you've set up this like 10,000 times discrepancy in the proton gradient, and these protons want to flow back into the cell. So this transporter utilizes the flow of protons flowing through it as the driving force to move some other solute at the same time against that solute's concentration gradient. Right? So if you look down here, so we said an analogy of a direct transporter might be, for example, this car jack, right? And the energy of this guy pushing on the arm can represent the breaking of ATP, right, to drive some other formation of a new bond, and that changes the shape of the jack. Right? Remember, even though this is quite obviously a car jack, and it looks pretty specialized for being a car jack, right? The reality is, even if there was no car or truck in this picture, and this guy was still working the lever over here, you would not say that this jack no longer works, right? Because the jack itself is still doing what it needs to do. It changes its shape, right? So that's a nice reminder that a lot of these ATPase pumps are just called ATPases, right? Because they just do what they do. And the fact that some of them move protons, some of them move other ions like sodium or potassium, that's almost like evolutionarily a second byproduct, right? Indirect over here is like the water wheel, because the water had to be raised up by some other pump somewhere that you don't see in the cartoon. And now that the water falls over the wheel, inside the wheelhouse, you're doing work, right? So outside the wheelhouse, you see the water splashing on the wheel and it's turning, and the gears inside allow you to do something like lift this heavy bag of grain or something, right? And move it around. Importantly, right, the amount of force that's needed to lift this heavy bag of grain and move it around, right, has to be less than the force of the water coming over the wheel, right? That makes sense, because if the bag of grain was too heavy and it actually requires more work to lift it up than the force on the wheel outside from the water supplies, it's not going to work, right? That's why up here, the force of the protons flowing through this indirect transporter has to be stronger than the resistance of this red solute being moved against its gradient, right? Okay. We then looked at the endomembrane system. In particular, we looked at examples like the endoplasmic reticulum, right, that surrounds the nucleus over here, and the two types of the ER, the rough ER that's studded with ribosomes, because that's where protein synthesis is made. And we looked at the smooth ER, right, where that's not where protein translation is made, but we have the smooth ER handling many different types of specialized functions. For example, right, in muscle cells, the sarcoplasmic reticulum, that's the special name we give the ER, smooth ER in muscle cells, handles calcium storage and releasing that calcium to allow your actin and myosin proteins in your muscles to contract. Right? We looked at how the smooth ER is also involved in detoxifying certain drugs, right? Like the CYP450 family of proteins that you'd find in the smooth ER that end up making small hydrophobic drugs more hydrophilic. So those drugs can actually be, more easily filtered out of your blood by your kidneys, right? So you detoxify your tissues that way. And we also said things like in your liver, the hepatocytes in your liver that store glycogen, right? They also would break down the glycogen through functions that are associated with the smooth ER. We also looked at the Golgi apparatus. We said as proteins are being translated in the rough ER, you would have little vesicles budding off of the rough ER that contains maybe these newly folded proteins, and they make their way to the cis Golgi network, right? The side of this Golgi stack of pancakes that faces the ER and receives newly made proteins in these vesicles is the cis Golgi network. Inside the Golgi, we learned how different sort of labels on the Golgi, on the protein products that are made, things like glycolipids, or things like, not glycolipids, sorry, small oligosaccharides, right? Short little polysaccharide chains can be added, right, to make these glycoproteins. And those little oligosaccharide chains can act as little tags that help those proteins as they move through the Golgi end up on the trans Golgi side, right, in specific vesicles that bud off of the Golgi that are heading to different places in the cell, thereby transporting those protein products where they need to go. Right, so this is the last slide for our review, and that's what this is going on here. We said that in the rough ER, a new protein is made, right, and certain short polysaccharide chains might be added to it, or oligosaccharide chains because they're short. Then when it moves in a vesicle to the cis Golgi network, it might be modified, so it might be phosphorylated in there. And as it moves through the trans Golgi network, right, it ends up associating with different parts of the membrane on this side that might have things like this receptor protein that specifically is the right shape to bind something like a phosphorylated manosugar that's part of this oligosaccharide chain, right? And that's what's being depicted on the right here. And as a result, you preferentially end up collecting this protein, right, in this vesicle that ends up being destined to go out to a particular spot in the cell, like a lysosome, right? So that this protein, which is meant to work in the lysosome, gets there, right, as part of the Golgi sort of packaging it up and sending it out to where it needs to go. Okay. All right. So that's our review. I know there's a lot of topics. And again, of course, even though I've touched upon many of the main points here, it's really important that you go back through the actual lecture notes to really sort of fill in a lot of the details in your understanding about these different types of topics, these different concepts. All right, so let's go ahead and look in the chat and see if there's any questions. So are DNA motifs the same thing as regulatory elements? Yeah, a better way of thinking about it is regulatory elements like enhancers, silencers, or insulators, right? Even though cartoons show them as like a colored rectangle or a colored block, I mean, obviously you all know it's not really like, oh, there's a purple block there or a green block there on the DNA. All it's trying to do is draw your attention to the fact that along that strand of DNA, right, there are certain short sequences, right, that represent specific DNA sequences, right, that are the right shape for key proteins to recognize them and bind to them. Right? So that short DNA sequence that's being recognized, right, is the DNA motif. Right? And so enhancers, silencers, and insulator regions would have examples of these DNA motifs in them. And that's how different transcription factors get recruited, right, to an enhancer or to a silencer or insulator, etc. Right? Let's see, if you ask us to describe the basic structure of a glycolipid, should we describe the simplest structure, lipid and sugar, the glycerol glycolipid structure, or the spin? Well, I mean, all three of them are glycolipids, right? So I would not say explain what a glycolipid is and then expect you to somehow magically know if I'm referring to a glycerol glycolipid, sphingoglycolipid, or a simple sugar group on a fatty acid. I think it's better for you to approach it like understand what those three different examples of glycolipids are that I gave you, right? What makes them different, and structurally, how you would be able to maybe recognize one as being different than the other. Okay. Question, is a transmembrane protein anchored in a membrane because a part of its sequence is hydrophobic? Yes, right? That's what I was saying, because if you were to unravel that protein, right, and just make unravel it into just a chain of amino acid residues, that's the primary sequence of the protein, right? That's the chain of polypeptide, or the polypeptide chain that was coming out of the ribosome when the ribosome was translating the mRNA, right? And then that chain folds up. But if you unfolded it, it would just be a big long chain of amino acids, right? And as you read along, what you would probably find is certain stretches of it tend to have more hydrophobic amino acids. Certain stretches of it have more hydrophilic amino acids. So hopefully now you realize once that thing is folding up, those different stretches of hydrophobic and hydrophilic amino acids will have a big impact on how this thing folds up in 3 dimensions, right? Because it's all surrounded by water molecules. Right? So on a globular protein, the hydrophobic stretches of amino acids would tend to be towards the inside, right? And the stretches of hydrophilic residues would tend to be sort of pointing on the outside. In a transmembrane protein, the hydrophilic residues would tend to be the ones on the sides of the membrane sticking out into the water, right? And you would expect that the amino acid residues that are going through the middle of the membrane as part of this protein, you tend to find more hydrophobic residues there. Okay? So a follow-up to this question is this the same sequence that becomes stuck in the membrane when the mRNA was being translated? It's not the exact same sequence, right? Because the truth is, whenever we're learning about examples of these sequences, right, it shouldn't surprise you that there are many, many examples of sequences that end up sort of happily being stuck in the middle of membranes, right? And those sequences are very specific to different types of proteins, et cetera. But the same principle applies, right? That you'd have a stretch of amino acids coming out of the ribosome that tend to be maybe more hydrophobic. And so as the ribosome is trying to get the polypeptide chain to translate into the ER, right, this sequence in the amino acids, when it gets to it, those tend to be maybe hydrophobic, right? So they'd end up getting kind of like stuck or stay in the membrane of the ER. And that would influence how the rest of the protein ends up being translated, right? And if that protein ends up being translated, so part of it is still in the middle of the ER membrane, right? Then you realize, oh, this is a protein that's meant to be some kind of a transmembrane protein, because then that part of the ER membrane, right? So let's go to this cartoon here. Right? That part of the ER membrane would butt off as a vesicle, but instead of having a free floating globular protein like in the cartoon here, you would imagine what we just talked about is you'd have transmembrane proteins sort of stuck to the membrane here, right? And they would be brought to the Golgi, packaged up, maybe put little labels on them, and vesicles would butt off of the Golgi, still having those transmembrane proteins as part of them, right? And maybe these transmembrane proteins are meant to be on the outside of the cell. So those vesicles are destined to make their way out to the cell membrane of the cell, right? The plasma membrane. And once they fuse with the membrane out there, what happens to these transmembrane proteins? You know, they just become introduced as part of the outer cell membrane, right? So hopefully that answers your question. Okay. Do we need to memorize structures besides lipid classes? For example, do we need to know the structure of lactose or mannose? No, you don't need to know the structure of lactose or mannose, but hopefully if you see a little ring of carbons, you'd recognize that, you know, it's maybe something like a sugar, right? Like, for example, you don't have to be able to draw this mannose structure here, but I would expect that if you saw something like this, right, you would not say, oh, is that a lipid, or is that a protein? And you certainly would not say, is that an amino acid? You would say, oh, that's a sugar, right? That's what I expect you to be able to recognize. Okay, are the hydrophobic regions on protein sequences lipids? No, right? Proteins are not lipids, right? Proteins can join up with lipids to form things like, you know, a molecule that has part protein and part lipid coming off of it. But remember, hydrophobic regions on proteins refer to the amino acid sequence. So go back to the first third of our semester, right? Why did I have you memorize the 20 different side chains of the 20 amino acids? It's so that you could realize that some of them are hydrophobic and some of them are hydrophilic, right? So if you string together a whole bunch of hydrophobic side chain containing amino acids, right, you have a region of the protein now that is hydrophobic. Okay. Oh, I guess you knew that because you said, or do we just know that in general they are hydrophobic side chains? Yes, right. So yes, in general, when I'm talking about the portions or domains of a protein that is anchored in a membrane, I expect you to realize that the amino acid sequence in that particular protein is most likely enriched in hydrophobic side chain residues. Right? How are the protein pumps attached in the membrane? Well, hopefully you already know that, right? How are any proteins floating in the membrane? What have we just been talking about? Proteins sit in the membrane because they have hydrophobic regions in them. So as the protein starts floating out of the membrane and those hydrophobic regions start getting exposed, like the membrane, like the protein's peeking out as it's coming out of the membrane, what happens? All the water just shoves those hydrophobic residues back in. And so that's why the protein doesn't float up and away from the membrane, and it doesn't float down and into the cell, right? If the protein is just the right shape with just the right amino acid sequence, it'll go ahead and sit in the middle of the membrane, right? Okay, so that applies to any protein that's a transmembrane protein, right? Transmembrane just means it goes across the membrane and it stays there, right? Anchored to the membrane. But I think that's a very good question though, because that's one point that I want you guys to realize, right, is that the membrane, remember I keep saying the membrane is nothing more than a whole bunch of hydrophilic and hydrophobic interactions, right? The phospholipid bilayer, all the phospholipids are arranged with their hydrophilic heads up, right? The polar heads up and the fatty acid chains down, simply because all the water molecules make them that way. Right? And likewise, all the proteins that are meant to be part of the membrane, like protein pumps, right, stay in the membrane, right, simply because of competing hydrophobic and hydrophilic interactions. That's why these protein pumps don't float out of the membrane, right? I think maybe one of the reasons why it's so difficult for students to understand that is it just sounds too simple, right? The fact that your cell literally is held together because water is pushing these molecules in a certain way, right? And the polar, nonpolar, hydrophilic, hydrophobic nature of these molecules, right, ends up determining how they stay, quote unquote, anchored. Okay? So I think anchored is kind of like a misleading term, right? Because you think of something being anchored as something being very solidly placed there. You know, I use the example of like the surface of our membrane, since it's very fluid. Think about a bathtub, right? And you fill it up halfway with water, and you have things like a rubber ducky, maybe a little toy tugboat or something floating in there, right? You don't imagine the rubber ducky or the little plastic tugboat somehow magically floating up into the air away from your bathtub, right? Nor do you imagine them on their own suddenly sinking underneath the water surface, right? They are essentially anchored to the surface of the water, right? Now, in that case, it's different because that doesn't do with hydrophobic, hydrophilic. That just has to do with gravity and buoyancy, right? But think about the proteins that are anchored to a cell membrane, right? Similarly, not floating up and out or floating down and in, but in this case, right, they're held there just by competing water forces, keeping them there, right? Okay. Where do all the phospholipids come from in our cells? So that's a very good question, right? So we get a lot of lipids from our diet, right? So the lipid component or the fat component of our diet has a lot of phospholipids in it already, because think about it this way, right? Anytime you eat something that's, you know, natural, whether it's plant-based or animal-based, right, you're eating their cells, right? I know that sounds gross, and I know it sounds on one hand obvious, but have you ever thought about that, right? You're eating organisms' cells, right? So you're getting phospholipids, right, by the billions of cells that you're eating, right? If you eat a steak or if you eat a salad, right? So those phospholipids get broken down in your body and they can be used as the phospholipid building blocks of your own cells. But other types of lipids also. We have proteins in our body that when we digest other types of lipids, for example, the omega-3s that we talked about that come from things like fish or fruits and nuts, things like that, we have proteins that can take those types of fatty acids and work on them to make phospholipids for our cells. So it's always a combination. We have cells in our body that are always being recycled, right? So when they get recycled, you know, those phospholipids can be used to make new cells. But we ultimately rely upon a constant intake of macromolecules in our food, right? Not only things like lipids, right, but of course proteins that provide us amino acids to build more proteins, right? Sugars, which is pretty obvious, right? Sugars that give us glucose that our cells can use for energy. And also probably the least obvious, which I think is interesting, right, is nucleic acids. Because just what I just said, if you're eating billions and billions of some organism's cells, right, whether it's something like cow cells from a steak or lettuce cells from your salad, right, they also contain DNA, right? You're eating cow DNA or you're eating lettuce DNA, right? And so your body breaks down the DNA and uses the individual nucleotides as raw material so that your cells can then put together the DNA in a way that you need it, right? So it's no longer cow DNA or lettuce DNA. It's been disassembled into monomers, right? The individual nucleic acids, and then reassembled by your cells into the sequence of DNA that your cells need, right? So yeah, just think about that, right? Any of you who have looked at a nutritional label of some product, you always see nucleic acids somehow listed on there also, but maybe you don't think about that, right? Anyhow, so, all right. So I think that's it for the questions today. All right, so good luck studying, and don't forget to log in on Wednesday at 10:30 to take the multiple choice La Lima part. All right, have a good day. Bye-bye.